To begin our next session, Emerging Therapies, and we have two outstanding speakers with us today who bring a wealth of experience and dedication to the sickle cell community. First, let me introduce Dan Bardos. Dan is the Global Medical Liaison for the Rise Up study at Agios Pharmaceuticals. His role involves establishing connections with trial sites, healthcare providers, caregivers, patient advocacy groups, and other stakeholders within the sickle cell community. Prior to his current role, he was a researcher focused on rare inherited arrhythmia syndromes and other cardiovascular diseases. Living near Denver, he enjoys the mountains and outdoor activities with his wife. Following Dan Bardos will be Dr. William Engelman of Fulcrum Therapeutics. William Engelman is Fulcrum's medical director for the hematology group. He has 9 years of experience in clinical development and drug safety. William holds an MD from Temple University and did his residency in pediatrics at the University of Massachusetts. Prior to attending medical school, he worked as an engineer. Together, Mr. Dan Bardos and Dr. William Engelman will guide us through today's important discussion on emerging therapies. Now let's get started and dive into this insightful session.
Therapies, and our first company is Agios.
So first off, thank you so much, everyone, especially during Sickle Cell Awareness Month. I'm really excited to be here, and I appreciate the opportunity to present.
Wait, just 1 second. I'm sorry. Can you say what month is it again?
Sickle Cell Awareness Month.
I mean, the crowd should be going right now. Yeah. Yes. We're celebrating Sickle Cell Awareness Month. Okay, I'm sorry. And go, doctor.
Yeah, thanks. But like I said, I'm really excited to be here and share what Agios has been working on from our clinical trial programs, the Rise Up program. So our disclaimer, just keep in mind, mitapivat is still an investigational product in clinical trials and hasn't been approved by regulatory agencies for use in sickle cell. What is mitapivat? So mitapivat works by pyruvate kinase activation. So this may improve anemia and also may improve sickling of the red blood cell in sickle cell disease by a dual mechanism of action. So first, it acts on a particular molecule, pyruvate kinase or PK, in a process called glycolysis, which is very important for the energy production of the red blood cell in the form of ATP. So it works to increase the levels of ATP within a red blood cell, thus enhancing or potential to enhance red blood cell energy metabolism and may improve membrane integrity, deformability, and the longevity of the red blood cell itself. It also can impact another molecule within this process called 2,3-DPG, and this may reduce hemoglobin S polymerization by increasing hemoglobin oxygenation, and thus may downstream inhibit the sickling process of HBS. We have an ongoing clinical trial, so I'll highlight first the phase 2 results that we presented at ASH in December of 2023, so this past ASH. And then we'll talk a little bit about what's ongoing in our phase 3 clinical programs. So first off, the phase 2 portion was a 12-week study where we enrolled 79 patients, and they were randomized to receive 27 patients received placebo, 26 patients received 50 milligrams twice daily of mitapivat, and 26 patients received 100 milligram twice daily of mitapivat for 12 weeks. At the end of the blinded period, we noted that patients treated with 50 milligram twice daily mitapivat, 46.2% of those patients had an increase of hemoglobin of 1 gram per deciliter or more. Patients treated with 100 milligram mitapivat twice daily, we saw 50% of patients had an increase of hemoglobin of 1 gram per deciliter or more. So we also saw that patients treated with either 50 milligrams or 100 milligrams of mitapivat had less red blood cell breakdown or hemolysis, and it led to red blood cells that live longer in patients who received mitapivat versus placebo. Importantly, we also saw that the annualized rates of sickle cell pain crisis in patients who were treated with 50 or 100 milligrams of mitapivat was less than those who had received placebo. We have to keep in mind, though, the phase 2 portion of the study was not powered to really assess this endpoint, but our phase 3 portion, which is a 52-week blinded study, will have the annualized rates of sickle cell pain crisis as a primary endpoint, along with improvements of hemoglobin as well. Also, of the 79 patients that entered the study, 73 of those patients continued into our open label extension. So this was up to 216 weeks or 4 years. So it's about 4 years plus 12 weeks that patients have access to mitapivat and additional disease modifying therapy. For the phase 3 portion, as I mentioned, it's a 52-week study versus 12, and we'll talk a little bit more about that in a moment. The safety results. So here, this lists the most common adverse events that were observed in either treatment arm, so the 50 or 100 milligram arm, or placebo at 10% or more. So we can see the safety profile was consistent with what we have also observed in our previous clinical trials and other indications, pyruvate kinase deficiency, thalassemia, and then we also have other collaborative studies looking at sickle cell disease as well. And what we can say is that no serious adverse events were determined to be treatment related. No serious adverse events led to discontinuation, dose reduction, or death as well. So overall, a consistent safety profile with what we've seen in our previous clinical trials, and we'll continue to assess the safety profile as well as, like I mentioned, the hemoglobin response and the pain crisis in the phase 3 portion of Rise Up. This slide highlights, where are our trial sites for phase 3. This highlights our sites in the US. So we have 30 sites that are active and enrolling in the US. So I'm sorry, we have 26 sites technically that are active and enrolling in the US. And this lists the regions. So in California, Los Angeles, San Diego, and then the Bay Area, we have sites that are actively enrolling, and then across other regions throughout the United States. We also have sites open in 16 additional countries. So from South America, Canada, and then the Middle East, Africa, and Europe as well. So it's a global study, competitive enrollment, and we're planning on enrolling 198 patients in the phase 3 portion. The enrollment criteria, so age 16 and up, and then the diagnosis criteria, if you have a genetically confirmed diagnosis of sickle cell, encompassing all genotypes related to sickle cell. So as Dr. Witten, Sherney mentioned, so HBSS, HBSC, HBS beta 0, and other genotypes are included in the Rise Up portion. Sickle cell pain crisis, two events that required medical intervention or hospitalization, to 10 pain events in the past year, and hemoglobin level between 5.5 and 10.5 gram per deciliter. You can also be on hydroxyurea therapy to be considered in the Rise Up study. However, your dose has to be stable for the previous 90 days prior to enrollment. Other exclusion criteria is, so if you're taking other disease-modifying therapies such as voxelotor, CRIZ, or L-glutamine, you cannot enroll if you've undergone gene therapy or have had prior bone marrow or stem cell treatment and other issues listed up here on the slide as well. So with that, I want to thank you for your time today, and we're looking forward to completing our enrollment for phase 3, and hopefully by after the blinded phase of 52 weeks, we'll have exciting results to share in the future. So thank you.
Thank you so much for that. So 2 things I just want to call your attention to the Rise Up. And I want to put in a shameless plug for Demetrius Wyant, who has a rap called In the Name of Sickle Cell. And if you haven't heard it, you need to hear it. And second, I want to kind of be disappointed about you giving me flashbacks to medical school and glycolysis and wondering if those first 2 years of medical school what was that for, you know?
Right.
And at the end of that is pyruvate kinase, and you know, and it's just
Yeah,
I had to memorize all that stuff. Now it makes it kind of,
yeah,
it's good you know it.
It's basically, in short, it's a complicated chemical pathway that's important for energy and the overall metabolism and the longevity of the red blood cell.
Thanks so much. Okay, Bill. Dr. Bill or Dr. Bill Engelman is going to now.
Is my mic on? Excellent. Yeah. So
from Fulcrum, and you got a rep there. Just come on up.
I like to move around and use my hands. You know that about me. I do. Yes. Yeah. Yeah. Okay.
Well,
So yeah, I'm gonna walk around.
You
Yeah.
have 10 minutes.
10 minutes. Well, let me get.
Oh, you can use
Let me get the
mine.
clicker. Is there a
If you wanna
clicker?
walk around, you can use mine.
No, I got my mic right here. This is what I want right here. Right? Are my slides up? There they are. Thank you, everybody. Thank you, Kind Wellness, and everybody for allowing me to come here today. My name is Bill Engelman. Everybody calls me Dr. Bill. I'm here with Fulcrum Therapeutics. We have a current drug that we're in clinical trials, phase 1 clinical trials I'd like to talk about. The drug's name is Pociredir, but let's first talk about us. Fulcrum Therapeutics. We're a pretty small company. We're about 70 to 80 people located in Cambridge, Massachusetts. We've only been around since 2015, so not even 10 years. We're about, like I said, 70 people, so we're small and mighty. You know, we like to, though, our aim is to developing disease-modifying treatments that result in meaningful outcome for patient, caregivers, families, and medical teams. What we're looking to do is get therapies that get to the root cause of the problem and not just to treat symptoms. So that's our goal here. We also can see here what's in orange. We take great pride in being purposeful patient partners who do this work, not just for patients, but with patients. You know, we want to speak to you and figure out what you need, and we're trying to do that. And I'll explain a little bit more about that in the upcoming slides. Here's our leadership team. Mel Hayes, he's our Executive Vice President of Patient Experience. Danielle Branch, she's not here today, but you'll see her here over the next few days. She's our Director of Patient Experience. And there's me. I'm the Medical Director of the Hematology Group at Fulcrum. I'm the one that's the Medical Director on this clinical trial running up, and I'm the one who can answer all of the scientific and medical questions on it. What we look to do, right, is to develop meaningful treatments. You know, we seek to develop medicines that can bring meaningful outcomes for as many people as possible. Like I said, you know, our goal is to get to the root cause so that we can improve signs and symptoms of sickle cell disease and improve quality of life for people living with sickle cell. In addition, you can see here, in addition to the science, we want to build lasting partnerships, strive to understand the patient experience, and develop impactful programs. Sickle cell is a lifetime disease, and we want to make sure that we're taking into consideration the entire life of a sickle cell warrior as we're doing this clinical trial. So in order to discuss where we are right now with our clinical trial, we have to talk about the phases and in clinical development for a drug. And there's a lot of words on here. I'm not going to go through all these, but I do want to, the way I like to think about it with the phases going through clinical trial, imagine this right here, where I'm standing right here. This is a phase 1 clinical trial. You know, this is done to look at safety, mostly. It is done to see if the drug is effective, but it's mostly done to look at safety. So phase 1 is over here. I'm going to come all the way over here on the other side. Here's a phase 3 clinical trial. Phase 3 over here. These are done mainly to look to see if the drug works. Is it effective? It's also, if you can see, I'm closer to the exit over here. Phase 3 is closer to the exit. This is the place where all drugs want to go. We want to go outside and into the real world so we can help patients out there. This is the phase 3 over here. Phase 2 trials are also a lot more patients. They're a lot bigger, like hundreds of patients, last long, a year or two. It's looked to see if it's effective here in this trial. Phase 1 over here, as I mentioned before, they're small. We're looking to get a few patients, maybe 20 to 50 patients over here. They don't last very long, a few days to a few months. Like I said, the goal here in phase 1 is safety. It doesn't mean that we don't look at safety over there in phase 3. We do as well, but those are designed to look mostly at how effective it is. And it doesn't mean we don't look at the effectiveness over here in phase 1. We do, but it's mostly to look at safety. And that's where we are currently today with this drug. Now, I also want to mention too, imagine, a lab behind me here. And the lab right here is where the drug comes from. It starts off in the petri dish and in animals. And it has to come out here into a phase 1 to start. So these are first in human trials out here as well. And what we are looking for our phase 1 trial here are brave warriors and patients that will help us move this therapy that we think is going to be very effective forward into this clinical trial. So we're looking for those few brave people that will help us do that. More about the study here. As I mentioned before, we have a phase 1 trial. Safety and tolerability is what we're looking at. We also want to look at to see if it increases fetal hemoglobin. I'll speak a little bit more about that. I'm sure most of you in here know about fetal hemoglobin. I'll speak a little bit more about that later. The trial itself is about 15 to 16 weeks. But what does that include? So that includes for a warrior that comes into our trial, it's a 12-week treatment period. But there's also a few weeks beforehand to look to see if they can come into our clinical trial. So there's some exams and tests being done. And then there's another 4 weeks afterwards, where we're looking to see after that drug stops, after 12 weeks of dosing, is that are there any residual effects as it washes out of the body? I'm going to skip over this because I'm going to talk about it a little bit later, but let's talk about what pociredir is. But in order to talk about pociredir, we have to talk about sickle cell, right? Right? People have seen me do this. This is a red blood cell. You can see it's round and it's flexible. And it has to be flexible because it has to fit through all of the small blood vessels in our body. Now, inside of every red blood cell, there's a particular molecule I'm sure you've heard of called hemoglobin. Now, hemoglobin, the job of hemoglobin is to deliver oxygen from the lungs to the rest of the body. If this is what most hemoglobin looks like, here's what a hemoglobin in a sickle cell warrior looks like. Can everybody see the difference between these two? Can you guys see that? No? See it back there? Yeah, excellent. Okay. Now, what happens with the hemoglobin in sickle cell is that they bind together. And I've heard somebody say, oh, so they form a chain when they bind. No, they don't form a chain. Because a chain, when I think of a chain, I think of something that's very flexible. It forms a long, stiff rod. That's called polymerization, right? It's stiff, right? And imagine that being formed inside of a red blood cell. What's that going to do to the red blood cell? It's going to misshape it, right? It's going to get that sickle shape, the classical shape. And they said that's stiff, right? Now, there's a specific kind of hemoglobin that all of us have produced in our bodies at one point in our lives. It's called fetal hemoglobin. And as the name, name implies, right? We produce this when we're fetuses, when we're developing in our mother's belly. Here, this represents fetal hemoglobin right there. I know it's my binky. Now, what we know is that if we produce enough fetal hemoglobin, 25% or 1 in every 4, what that does is that prevents these hemoglobin molecules from sticking together, prevents that polymerization. So if we can get enough fetal hemoglobin produced, we're going to produce red blood cells that go back to that round, flexible shape. And like I said, we have that machinery inside of us to do that. So that's what we're trying to do here with pociredir is produce fetal hemoglobin, right? aims to produce production of fetal hemoglobin. Production of increased fetal hemoglobin reduces symptoms of sickle cell. Our drug is a once-day pill. We have, right now for this trial, it's 2 capsules you take once a day, but we're trying to form it into a single pill once a day. And that's what we're doing right now with the clinical trials to try and test this, especially the safety to move forward. And I'm sure most of you have seen stuff like this before and know this. And what happens in sickle cell disease is disease that affects the body from head to toe. Increased stroke, increased problems in the lungs, such as pulmonary hypertension, acute chest syndrome. Nephropathy, that's a fancy term for kidney disease. Osteonecrosis, that's another fancy term for bone disease, as well as ulceration pain. Now, we do know that if fetal hemoglobin levels get up, it's at around 25, 30% or higher, these symptoms go away. These symptoms here go away if we get up that high. So that's our goal. But let's talk about how pociredir works. I like this slide. It's very complicated, but I really like this slide. Now, I want everybody to look over on the left side where it says PRC2 complex right here. But look right underneath it here. This is where I want everybody to concentrate. Right underneath it, what looks like a piece of string that's wrapped around a couple of spools. Okay, that piece of string is DNA. And DNA, I'm sure we've all heard this before, that's the blueprints of our body. That's what's needed to produce the proteins, the blueprints of our body. Now, these spools, they're called histones. And if DNA are the blueprints, think of histones as being the file cabinets where that DNA, where those blueprints are being stored. So the histones are the file cabinets. Now, this red guy, this red cloverleaf looking guy here and there, that's called a trimethyl group. The name's not important, but think of that as being a lock that gets put on those file cabinets. Not this particular piece of DNA right here has a gene on it called HBG. And not long after we're born, this guy here, this PRC2 complex, comes in and puts the locks on those file cabinets. By putting the locks on those file cabinets, that HBG gene is not being read, and therefore we stop producing fetal hemoglobin. Because when that HBG gene gets read, the downstream effect is to produce a whole lot of fetal hemoglobin. What our drug does, pociredir, is it comes in and it attaches itself to this PRC2 complex at this position called EED, but that's not important. But when it attaches it to itself, what it does is it prevents this trimethylation. So it prevents those locks, stops those locks from being put on. And so what can happen is that DNA can unwind. You know, the blueprints get taken out of the file cabinet. They're being read. That HBG gene gets read, and we see a downstream effect of producing a whole lot of fetal hemoglobin. The other thing you can see here, it says it's highly selective and clean off target. What that means is that we're only seeing it, work in this one spot in the body and nowhere else. And that's important because that means we're going to see fewer adverse events because we're only seeing it work in that one spot. So we're very excited about this. Here's some data, some early data from this phase 1b trial that I'm talking about. How you read this. I like graphs. I know I'm kind of a nerd that way, but I really like graphs. But let me explain how you read this. Absolute percent fetal hemoglobin change from baseline. How we read this is the amount of fetal hemoglobin that gets produced and made on top of what a warrior is already making prior to taking the drug. So let's pretend like, the absolute change here is 10, and a warrior makes 5% prior to taking drug. They're now making 10 plus 5, which is 15. The warrior makes 10, and that absolute change is 10. Now they're making 10 plus 10, or 20%. So that's how that's read. And what we're also seeing here, this number of days on drug, and I told you it's a 12-week treatment period. That's the dashed line right there, 12 weeks. What we're seeing is dose dependent. What that means is that as we're increasing dose, we're getting more fetal hemoglobin getting produced. 2 milligrams here after 12 weeks, you have about 3 to 4 above baseline. The orange here, that's 6 milligrams. We have near 8, maybe 9% added on. And this is the data right here. This graph here in black. We only have preliminary data here. This is after about 4 to 6 weeks of treatment. But after 4 to 6 weeks of treatment, we got about the 10% range. Now, imagine this going all the way up out to the 12-week points. We can easily see ourselves getting up to almost the 20% range. And remember I said before, it's 25% we're looking for. And you can imagine if somebody's making even a little bit of fetal hemoglobin and we're going all the way out and it's getting about the 20% range, we can get into those ranges where sickle cell warriors are asymptomatic. We also don't have any data on here for the 20 milligram dose. We have a higher dose we're doing as well. We expect that one to show even higher levels of fetal hemoglobin get produced. So we're very excited about this. So who are we looking for to come into this trial? Of course, we're looking for warriors who are willing to, fill out the paperwork and willing to volunteer for this study.
Hey, Bill, 2 minutes.
2 minutes, got it. Adults, ages 18 to 65, looking for genotypes of SS, S beta 0, and S beta plus. Now, we know there are SC patients out there, and we are looking to include those. We currently do not have that, and I can't promise that we will, but check back because we're looking to include SC patients in the study as well. Patients have to have tried hydroxyurea, and either it doesn't work or they can't tolerate it in order to come into the trial. They also have to try one of the other medications on the market, such as voxelotor, crizanlizumab, L-glutamine, have tried it and either it doesn't work or can't tolerate it. Or if you can't get it because it's too expensive or other reasons, you can also be eligible for the study. You cannot be on hydroxyurea for the study, unfortunately, though. So you can't be on hydroxyurea. There are other criteria as well, but these are the big ones and the main ones. Here's where our study is currently taking place in the United States. We're going to have about 20 sites open in full. But here in the United States, we have one right here at UCLA, just north here. We also have Boston Medical Center, the Bronx, New York. We have Wally Smith site at VCU. We have University of North Carolina, University of Miami. We're also looking at Tulsa, Oklahoma, Mississippi, as well as we're about to open a site in Baton Rouge. We also have a site in South Africa, as well as we're opening some in Nigeria soon as well. This is important. We are providing a lot of support for this study. So we will pay for transportation, planes, trains, and automobiles to the site, as well as meals, hotel room, or gas to fill your car. And that's not only for the warrior that comes in to volunteer for the study, but that's also for a caregiver that comes in. But I want to point out over here on the right, wage loss and childcare. We do know that to get involved in a clinical study, it requires a lot of time and effort. We will pay for any wage loss for taking time off from a job, as well as childcare that you would need to come into the study. And here's some information if you want more. I'm also sitting back there, and we will be around here. Folk will be around here for the next couple of days to speak about and answer any questions. Thank you. Thank you.
Bill. Bill. Thank you for bringing your Tinker Toys.
Oh, absolutely.
And I always talk about how important it is for warriors to understand what's going on in their bodies and what medications do. And I think you did a phenomenal job of explaining what actually happens, So thank you for that. And I just have to make one other comment, and it just has to be that the compensation, just paying attention to what it involves for someone to be involved in a clinical trial and for you to, take care of that is just so meaningful. So thank you for all of that.
Thank you.
Okay. Now we have questions and answers, and we're going to go first in the room.
Yep, we're going to go first in the room and then virtual. And for our virtual attendees, if you have any questions, please make sure you put your questions inside of the question and answer part of the CVent app.
Somebody in the room has to have a question, right? Even if it's, where can I get those Tinker Toys?
We got the question.
Yes. Tell us who you are. And somebody going to bring her a mic? Or thank you.
Thank you for the proposal. Hi, I'm Juliet Romeo. I'm a sickle cell warrior SS. My question is for Dr. Bill. I'm very excited about this study, and I would like to know about if you are on blood exchange, what is the process?
Oh, unfortunately, we are not taking anybody who are on transfusions or blood exchanges into the study.
Any other question?
We got to go to the virtual first, or do we?
Nope, you could go to another in-person question.
Perfect.
So I want to know if clients can self-refer, or does the doctor have to refer them?
Self-refer, I'm not quite sure I understand the question.
Can they make contact and
oh, they can certainly reach out to the doctor,
Okay.
right? And if I can give you a flyer here. We do have a website that if you fill out a form on the website, we will actually contact you, have one of our patient liaisons contact the warriors and direct them to the closest site or the site that'd be most convenient to them.
And Pat, for the 1 or 2 people in the room that don't know you, could you please just say who you are?
I'm Pat Corley, and I'm actually probably the person that's been in the sickle cell space since we first organized the 10 comprehensive sickle cell centers, which was funded by the NIH. And so I am currently just doing a lot of work with Cayenne Wellness and International Association of Sickle Cell Nurses and some other organizations out there, and partnering with whoever there is in the country that needs help.
Thank you, Pat. Who else has a question for us? Oh, I'm gonna give her.
We can take another question in person.
I'm Wanda Williams, sickle cell warrior. And I'd like to applaud you for at least upping the age to 65 for people that you're considering, because usually people stop at like 30 or something like that. So compliments to you for that. And is there any interest in increasing your age, or is there a reason why you would not go beyond that?
Is there?
Dr. Bill.
Well, thank you. Yeah. One of the issues we have to always weigh in a clinical trial are the comorbidities that come along with patients that come into it. And past 65, oftentimes there are comorbidities as well as other medications that the patient's on that may interfere with the results of the study. And that's pretty standard. That's not just for a sickle cell trial, but that's standard across most clinical trials. Yeah.
Okay, so last time I announced a break, it was a little premature, but I think we're ready for that. Before we do that, Carolyn, Dr. Rowley. Okay. Okay, so the only thing that I'd like to do is that if there are, I know there are a lot of warriors in the room, and if you'd like to be acknowledged and are comfortable standing, I'd like for you to stand and be acknowledged, because it's all about you. That's why we're here.
Look at all the wonderful warriors. I mean, clap a little harder for the warriors. Let's go, warriors. Woo! And to all of the warriors that are watching virtually, thank you also for joining. You got a hand clap too.